Thanks for accommodating me virtually. I really appreciate being able to join, and I hope to dovetail a bit with the remarks that you just heard from Tim. So what I will do in the next 25 minutes and have some time for questions at the end is talk about some of the progress in product approvals over the past year, talk about what we're doing to advance the regenerative medicine framework for cellular therapies and tissue therapies, talk about what we're doing to expedite gene therapy development, and really try to look forward toward in vivo genome editing as an example of how we can really lean into the regulatory process. This past year, we had some breakthroughs in the regenerative medicine therapy area. We had the first of the TILs that was approved, the first tissue engineered vessel approved at the end, towards the end of the calendar year last year, and the first mesenchymal stem cell therapy that was approved. And it is true, you could say that some of these had a long development time, but they made it. And I think it's exciting to see that hopefully now that the path has been essentially plowed, we'll see other products starting to come up behind these. Additionally, if you look, 2024 was a good year for gene therapy approvals. You can see starting with an additional indication for Casgevy and moving on through, we have had a variety of gene therapies approved this past year, ranging from hemophilia B, sickle cell disease, thalassemia, that crossed over the 2023-2024 years. And also the first of, as was already mentioned, the first of the solid tumor T cells. So we now have really in blue here, you're seeing this group of T cell therapies, most of them CAR T cell therapies. And this is now no longer now a onesie or twosie. This is now becoming a workhorse of hematologic malignancies. I think we'll soon see it become one of also a workhorse for perhaps autoimmune therapies, as we're starting to see data coming out of those trials. And so this is an exciting place to see things. Last year, there was this issue of possible concerns with the T cell lymphomas with these CAR T cells. I think this year we are feeling reassured in this regard, and I think you can assume or you can take it that in the not too distant future, you'll be hearing from the agency as we kind of refine the warnings that we made last year in this area, taking into account the data. Now, this is a great example of how in new fields, things can get really complex quickly, because it turned out that what we were witnessing when we were witnessing these T cell malignancies in the setting of CAR T therapy was a combination of a very small signal that was likely related to the actual CAR T and a actual background of secondary T cell malignancies in people with B cell lymphomas. So really had to be deconvoluted, but I think, again, we'll adjust to that. Probably some lessons learned there for the future, but I think this is an exciting place here. I think it also makes us feel reassured as we see these products moving into the space for autoimmune disease and other diseases that are not malignancies. In terms of the gene therapies on this list, The green products here, the genetically modified stem cell therapies, I think here we're starting to see these make their way through. I think this also brings up a really interesting set of challenges that have had to be overcome. We already heard about the issue with reimbursement. I think there are also the practical issues of getting patients through these therapies because there's a lot of complexity in these patient populations. And now that that's being addressed, I think everything from fertility issues to withdrawal from narcotics for sickle cell patients who are essentially cured of their disease through these products. A lot of these things have been addressed now, and we'll see these, I think, hopefully come further into use. And hopefully we'll start to see second generations of these products start to evolve as we get to places where cytotoxic conditioning regimens will not be necessary, hopefully, at some point. We'll have some alternative as these products evolve, because that is one of the things that probably is limiting their use at the moment. And then for the products in purple, well, the directly administered gene therapies continue along. AAV continues to be the workhorse here. And hopefully we will continue to see advances in AAV vectors with hopefully better generations of vectors that create less immune response and also better manufacturing technologies which are in process. I think one of the things that I'll talk about later that looking forward to is that the issue with AAV continues to be that it is a process where the manufacturing is heavy on traditional biology, messy biology, and not so much the kind of synthetic processes that we've seen with, for instance, lipid nanoparticles and mRNA technology. So this is just an area that has been a challenge, and we just remains to be seen whether we can advance the AAV manufacturing to really move the field forward even faster. I just want to turn now briefly to an issue that is something that we've been dealing with for some time. It's a little bit of an asterisk because it's not a genetically modified issue, really, but it's one for basically cellular and tissue therapies. We have human cells, tissues, and cellular tissue-based products, or HCTPs, which comprise a variety of different tissues or derivatives from those tissues. We've really only come to regulate these in the 21st century. The regulations started to be put forth in the late 20th century. And the regulatory framework basically to not over-regulate created or under-regulate, tried to create two regulatory tiers. One, which are kind of the products are regulated like traditional biologic products, and the other products which kind of have their safety and effectiveness inherent in what we know about those products, as long as they're prepared in a manner that prevents the transmission of infectious diseases, is in a different tier. So we have these what are called 351 HCTPs and 361 HCTPs. The 361 HCTPs don't require pre-market review and approval. The tension here is a natural one then. You would much rather have something be a 361 HCTP than a 351 HCTP because of the wide gulf in the requirements. Toward that end, we've issued a fair amount of guidance. These guidance documents issued in 2017 and then updated in 2020 to try to help people distinguish what products belong in which category. But the issue is the complexity of these products has continued to evolve, and this simple framework of two tiers is not optimal. And that's in part because what we have here is a really broad array of products where they're very different in their complexity. And when you actually plot them out, many of them are really simple to manufacture. There's not a lot of issues in their manufacturing, whereas others are very complex. same thing in terms of clinical efficacy. Some of them, the clinical efficacy, we know from long standing historical data what these products do, like an irradiated skin graft. On the other hand, genetically modified stem cells, those really require a fair amount of clinical data. And then there is a whole bunch of things in between. Mesenchymal stem cells used for orthopedic purposes, allogeneic pancreatic islets. And I could have busied this up with a bunch of other things, but since I know how large the room you're sitting in is from being there in the past, I figured I wouldn't make it difficult for the people in the back of the room to see this. But just the basic bottom line here is that we now understand there's a real complexity here. And given that broad spectrum of cell therapy products, we really need to think about ways to better get them into a regulatory framework. And what's happening here is that self-designation is not really working as well as we would like it to. Some are self-designating so that they are under-regulated, and other products we're clearly over-regulating. Example might be pancreatic islets. And so we are exploring a risk-adapted framework based on carefully considered variable manufacturing and clinical data requirements. And it's not fully formed. And in fact, we are going to want to hear from industry and academics on this. And there'll be a FDA scientific workshop on February 25th of this year to talk about this and to essentially start to plot a way forward, because we would like to relatively soon put together a proposal for a framework that would be more appropriate. So now I want to turn to kind of a last major topic bucket before coming to kind of an example of where we can come to in our regulatory work. Advancing rare disease products, something that is clearly something we were trying to do. In some ways, advancing rare disease products is a test case for us in gene therapy of what we could do in larger populations. If we can get it right in rare disease, hopefully we can get it right in larger populations. For the past year, we've had some areas that we've either had milestones in or are continuing to work in, where we've stood up a rare disease hub. We're trying to move forward accelerated approval, as you've heard, trying to work towards addressing the issue of regulatory differences between different jurisdictions, and trying to essentially apply best practices that could help move forward these products. And so the Rare Disease Hub is a collaboration between the Center for Drugs and the Center for Biologics, really trying to enhance collaboration and consistency across the FDA to address common scientific, clinical, and policy issues related to rare diseases. And what we're trying to do here is make sure that if somebody is using a biomarker in CBER, that they're not getting different information at CDER, for example, or at other parts of the agency. So trying to essentially harmonize and make it simpler for industry to get feedback. We've already assigned staff to this from CDER and CBER, and the hub director of strategic programs has been brought on board. That person is Amy Rick, and she is, I think, a wonderful addition. We have a strategic plan for the coming year that's been developed, and it's going to be released soon. It's focusing on 5 C's: communication, community coordination, creativity, and cooperation. Hopefully, this will really elevate things at the agency. It should make it easier also, particularly for patient groups, to communicate, because it's essentially one-stop shopping. Now, I think leveraging accelerated approval is something in this area that will benefit both rare diseases and ultimately all diseases. I think there is a lot of controversy. You can hear about it, about accelerated approvals, but I think the science inherent in the development of many gene therapies and really facilitates the use of biomarkers as endpoints that are reasonably likely to predict clinical outcomes. That's because we can use animal models or human observations to find levels of these proteins or upstream or downstream metabolites that are able to reasonably likely predict the clinical outcomes. And then we can come back with a study later on to transition an accelerated approval over a traditional approval based on clinical endpoints of how patients feel, function, or survive. This hopefully can help us get through a large number of diseases in a much more rapid fashion than we could otherwise. For some of the genetic rare diseases, particularly the neurologic diseases, where you have very little natural history, being able to leverage biomarkers like this and using animal models and leveraging all the human data that you can get really is a way that can hopefully bring some relief to patients in need much more rapidly than it would take to get long-term natural history studies followed by longer-term trials. So this is something we are working towards, and as Tim noted, we're enthusiastic about because this, combined with the work that can be done on platforms to advance various therapies, I think gives us a very powerful tool to get therapies to patients. One of the other things we're trying to do is help get regulatory review of rare disease therapies harmonized between different countries. We have a pilot ongoing currently with the European Medicines Agency. The idea here is to try to harmonize our requirements so that a single application could be submitted to multiple regulators and potentially be reviewed collaboratively by those regulators so that there is less rework. The potential to have essentially one-stop for these types of rare disease products could hopefully allow patients in multiple regions to benefit because what starts out from a non-commercially viable market in one country could suddenly become a very viable market if you are able to introduce it into multiple countries simultaneously without very much extra work. And there's enough work already that will be involved in obviously pricing negotiations, et cetera. But if we could at least get out of the way of in terms of the actual common technical document and the requirements, that would, I think, be a really great thing. So we're going to continue to really work on this and hopefully make some real progress in the coming year. We're also trying to apply the best of what we knew from Operation Warp Speed, which something I talked about previously, but it's now become a reality. Our support for clinical trials advancing rare disease therapeutics pilot. This is really the START pilot is something where we've now chosen the first 4 programs. We are working to make sure that there's essentially a constant avenue of communication with these sponsors. We're going to be evaluating this pilot with the help of an external consultant, both internally at FDA and externally through the sponsors that are participating. This slide basically shows that the 4 programs that were selected, these are all relatively severe diseases: NGLY1 deficiency, Rett syndrome, Canavans, and methylmalonic acidemia. And you'll see from the names of the companies here that this is a range of sizes. We wanted to see how this would assist a variety of different size entities. Hopefully, this will prove it's worth in terms of shortening the time of development. If it does, I think we will have the rationale for potentially incorporating this type of a program into our next set of user fee negotiations. Finally, I just really want to talk a little bit about platform technologies and their application moving forward. This concept of being able to reuse manufacturing information, toxicology information, is one that is very it's very relevant for gene therapies, where oftentimes we have these vector backbones that are reused over and over again. Now, it's a little tricky with AAV, perhaps, because sometimes the inserts of different sizes can lead to different outcomes in manufacturing, but by and large, it holds up. And we have the Omnibus Appropriations Act of 2023, which allows us to designate a given product, a platform, that allows one to use it as a reference as you come along with additional products. We are actually kind of extending this so that people in development, even if it's not an approved product, can have a group of products under IND progressing forward, essentially cross-referencing each other. But really what's important to recognize here is that as we move into CRISPR, we really have the poster child of a platform, which is here. You can imagine a editing technology here where you need to edit something in the liver that could be a blood disease, hemophilia B, or a metabolic disease, hereditary hypercholesterolemia, and you can have something like the prime editor on the right side of this slide. These prime editors and the delivery mechanism, you're going to have products that are 99% plus identical and will be manufactured the same way. This is really a platformized approach, and it really makes us have to step back and as regulators think about whether we need to now start to think about whether we can have a different approach to what we consider a product. In other words, our traditional way of approaching things is we have a disease and we have a product for treating that disease. Here, we're going to have something that is probably going to be more like a tool which will treat a variety of different diseases in a given location, provided those diseases have certain similarities. So I think this is an exciting place to move forward, and I think it's particularly exciting because there are now ways of delivering in vivo genome editing. We can have untargeted lipid nanoparticles which target the liver, but people are finding better ways to target the liver by actually studying those lipid nanoparticles. We have the use of cell surface molecules that can target T cells, specific lipids and cross-linking to target bone marrow. People are looking to target brain. You get the drift here. People are looking to see how far we can move this in vivo genome editing platform. Part of this that's very nice is that we know from the COVID-19 pandemic, another thing that came out of this is since the mRNA vaccines were our workhorses in the United States, we know how to make lipid encapsulated mRNA, and that mRNA can potentially drive CRISPR in these areas. Now, this doesn't work currently to every site that you want to get CRISPR at this point. The brain is a challenge still, but if we can exploit this as much as we can, this really transforms how we make products in this gene therapy space, because as opposed to AAV, which is a biologic process in every respect, you have to grow cells essentially and then infect them and get everything right. Here, this is much more of a synthetic process where you have template coming off of, it could be E. coli-based template or even synthetic template soon, and a process that can be actually almost miniaturized. That's what the example of this device is on the right, a manufacturing assembly for lipid nanoparticles. This could really transform the cost of production and the complexity of therapies. And I think what you really have here is something, I did not yet mention artificial intelligence, so that obligatorily I'm going to bring it in now. If you have this delivery vehicle, advanced manufacturing technologies which allow you to make these products essentially using continuous manufacturing or on-demand manufacturing. Combine that with artificial intelligence that's being applied to control the manufacturing process and to do things like control for off-target effects. And if we apply a flexible regulatory platform, I think all of these could lead us to accelerate progress here. So I think what you'll see is that we're going to actively advance the science and regulatory framework for gene therapy, as well as try to expedite the review process to really help bring the promise of genetic therapies to the broadest number of people globally. There have been setbacks over the past year or two. We know that. But I think we are now past the place of knowing that gene therapies can be a reality. And I think it's just a matter of trying to accelerate the pace of progress here so we're not doing onesies and twosies, and that we essentially are able to really open the floodgates for reaching the maximum number of people who could benefit from the products. So I'll stop there and really appreciate your listening today.
Hey, Peter. Peter, while we have you, we're generous enough to offer to answer a few questions. Maybe we could do sort of lightning round. We've got 3 that we'll do, and then after that, I'd invite Whitney and the panel to come up once we're done with these. So I'll ask the first one, then Linnell Hoke from BMS will ask the second, and Bob Smith, our chair, will ask the third, if you're okay with that. These are tough. We're not going to let you off the hook easy. These are tough questions. No, so I don't know if you heard, but you and I have talked about this. We've gotten a lot of great feedback on Nicole Verdun, the team she has brought in, her active management style. It's been very good feedback. Two-part question. This is the easy one. How do you think that's going? And then the second one, how is the team within CBER, within OTP, holding up just in general, given the changing of administrations? High-level thoughts on both.
So first of all, Nicole has brought a wonderful breath of fresh air and an approach which I think has helped diffuse throughout the office towards really leaning into the kind of development that we'd like to see, open-minded thinking, how do we get there, rather than how do we not get there. And so I think and that continues to grow as we bring new people into the organization. So that's really great. Really, in terms of how people are holding up, I think, pretty well, because I think the products that we're regulating are exciting. I think people see this as an important public health mission. I can't predict, what certain things will, certain actions may cause certain reactions. I just don't know what all the actions are going to be yet. But right now, things are going pretty well.
Great. Okay, thanks. I'll invite Linnell up just to do a follow-up question.
Peter, thank you so much. I know how incredibly busy you are, and so to accommodate time to give us a kind of window into how you see things evolve over time and how the FDA will continue to support these transformational medicines, and to your point a moment ago, and how we can get it to the most patients that can benefit from it is great to hear. My question comes back to CAR T cells. As you said, we now have 6 of those assets. They've been in the market ranging from 7 to 3 years, depending on the asset, depending on the disease area. But as we sit here today, there are many, many patients that could benefit from CAR T cells, but only 2 in 10 patients receive CAR T cells. And initially, that was always a manufacturing challenge. It was that manufacturers could not scale. Now that we see ourselves unconstrained, that 2 in 10 is not moved. Maybe you can comment on maybe how you see that accelerating or growing over time, and what we could be doing in partnership on how we actually address what will be a growing issue if we can only get 2 in 10 patients getting access to a modality that they can benefit from.
Yeah, so this is a great question, and I think this is a great place for partnership with ARM, FDA, others, to try to really understand what is going on here, because as a hematologist oncologist, having watched essentially barriers to application of novel therapies before, I think obviously there are certain things we can start to move forward. Like I think we can hopefully start to think about, for low-risk patients, how we can move towards better outpatient treatment, how we can help essentially a tier of hospitals that might not be the academic medical centers, but might be the next level down, be able to apply these therapies so that people can get them closer to home. So I think that's kind of a simple low-hanging fruit. But I think we're also going to have to understand what might be barriers to physicians wanting these to apply these patients. And I'm going to dare say something. I hope I don't get thrown out. my MD. They won't take it away from me, but doctors oftentimes like to keep their patients close to home and close to therapies that they know. And oftentimes for these therapies now, they're having to refer them out, and they worry that they are not going to come back. And so I think we have to figure out how whether that's a component of this. I don't know for sure that it is, but it has been an issue in the past. I was chief clinical officer of a cancer hospital, and we watched this in the past. So I think we need to figure this out. But that said, we will do what we can on the FDA side, which I think is clearly part of the issue. I'm just not sure that it's all of the issue.
Fair. Thank you so much.
Hi, Peter. Thanks again for joining us and sharing some of the innovative thinking and kind of the exciting approaches on the gene editing and gene therapy as a platform. Could you just share a little bit more about the scope of that, potential next steps, and also how you're interacting with other regulatory agencies like EMEA, MRHA, and PDMA in this particular platform approach? Because it seems like it could be significantly advantageous for both the agencies and also for sponsors.
No, thanks so much for that question. I mean, I see this as really potentially transformational for how we can regulate these products in a way that really could decrease cost and increase efficiency of getting products through. And so at least initial conversations, and I'll be transparent that they're not advanced conversations yet, but at least initial conversations show a fair amount of receptivity from EMA and PDMA towards this concept, because I think we all see the fact that if we have 10,000 rare diseases and we can only get 10 to 20 products approved per year through the pipeline, that's just not, we're never going to get there to cover these. And so we need something that will get us there faster and yet maintain the kind of quality, safety, and effectiveness that we'd like. I think this platform approach, particularly if we're applying it in a very thoughtful manner, and CRISPR perhaps gives us a way to do that, may let us get many more patients treated in a way that maintains all of that which we as regulators hold dear, but also allows us to move much more rapidly.
Well, great. Thank you so much, Peter. Really appreciate your taking time to join us this morning.
Thank you so much.